Hi everyone, welcome back. So already 2 o'clock, so I will proceed to the topic inheritance and genetic control topic of DNA replication. So for this part, is part 2 lah. So actually for the morning life, I already explained the experiment in the topic of DNA replication. So Fred Griffith experiment, Ellery, McNeil, and McCarthy experiment, and Hesher and Chess experiment, I already explained. So if you want to refer to these 3 experiments, you can refer to my last life. Okay, so for this slide, I will only concentrate on model of DNA replication. Okay, for your last life, actually last life, you already know the 3 experiment, the final experiment prove that DNA will carry genetic information. Okay, so DNA will carry genetic information. This is the final conclusion that made by the scientists for the previous experiment. So now, after we know the DNA carry genetic information, so we have to know how DNA replicate. Replicate, what is mean by replicate? So replicate is macam copy lah. So satu DNA, we know that satu DNA dia dapat macam ada double strand, right? Actually the DNA is not like this lah. And then actually DNA is like this. But as you know that the DNA is the molecule. So the molecule actually have the age. Ada umur dia. And then we have to renew our DNA from time to time. We cannot use only one DNA one DNA for the whole lifetime because it is molecule. There are it has the lifespan one. Every type of molecule they have their lifespan. Tak kisah. They have their lifespan. So they have to replicate more and more. They have to replicate to replace the old one. So replicate means they akan produce banyak lagi DNA lah. Satu DNA molecule dia akan produce banyak DNA lagi. So this is what it calls as DNA replication. So normally DNA replication is occur at the mitotic cycle. Don't know you all still remember or not the cell division cycle. I don't know you all still remember or not lah. I don't know. And we know that our first stage is there are many stages, right? And then the last stage is about the mitosis, right? And then there are S1, S2, and also got one stage is called as DNA replication phase. So inilah masanya untuk DNA replicate. Ini I think you in SPM you already learn. SPM no, you will see this term DNA replication, but you just know the definition only. Definition of DNA replication is to replicate to produce more DNA from the DNA. Macam kopi lah, macam dari satu DNA menghasilkan lebih banyak DNA lagi. Okay, ini DNA replication. Okay, lepas tu we have to know how DNA replicate. How? We already know DNA carry genetic information, so we have to know how DNA replicate. So there are the DNA replication model proposed by Watson and Crick. So I think these scientists you are familiar with it because the structure of DNA is proposed by who? Inilah. Same people. Almost of same people. Because structure of DNA is he proposed. And also, he also proposed the DNA replication model. So this is the model that he suggested that how DNA replicate. So there are he suggested total 3 replication model. So the first one is conservative, second one semi-conservative, and third one dispersive. Okay, so what are the difference? And only actually he just suggests, ini hanya suggest or propose lah, suggest or propose only. He also don't know. He also don't know how actually DNA replicate. He just suggests based on the theory he know. Based on the theory he know, he just suggests and propose only. But actually, which model is actual model for DNA replicate? He don't know also. He just suggested model. Okay. Okay, so what are the three model he suggests? The first model is about conservative model. Okay, so how DNA replicate? He just know that the Watson and Crick, he just know that the DNA, the double-stranded DNA, DNA is double-stranded, right? Double-stranded, it will split into single-stranded DNA. Lepas tu, each of these will act as template. It will split by breaking down the double bond. If you want to split down the DNA, double-stranded DNA into single-stranded, you have to break down the double bond and then become two single-stranded. And then they act as template. Yang kedua-dua ni they act as template. Template means what? Template means ini macam satu informasi lah, because we know that our DNA contain many nucleotide. So like many nucleotide like the nucleotide A, T, C, G. Dia ada macam ni kan. Betul ke? A, T, C, G. Betul. Yang ni this is a template, so the newly formed will become A, T, A, G and C like that. Ini act as template, yang atas ni act as template. And then this is the daughter. Stranded. Inilah yang hasilkan baru punya. So, yang ni dia tahu saja. Yang ni orang scientist ni, he know the DNA replication is something like that. So, he use this theory and then he will propose three model. Okay, what three model he proposed? First model, conservative model, he say that the two parental dissociate after acting as template for new strand, thus restoring the you will see this. This is the parental template, and then you see it will produce the two DNA. Akan produce two DNA punya. Satu DNA akan produce two DNA punya for DNA replication. But for this, yang ni, awak tengok sama-sama sebiji dengan yang mother, yang parental punya. Yang ni totally not same. So this means what? Means that when the template, the DNA is split into the single-stranded, then this is this one act as template, right? And then it will produce the daughter stranded one. Yang ni daughter stranded one. Lepas tu, tapi yang ni model dia cakap parental and parental will combine together and daughter will combine together. So totally you will get like this. Yang ni totally daughter punya, and another one is totally for parental only. And also like this. Yang ni. Why I use different color means that this is all are daughter strand. Daughter strand, this is the parental strand. The characteristic, the nucleotide base is totally different. This one, parental is totally same as the original one. And the daughter is totally different from original one. Why? Because the daughter stranded will combine with another daughter stranded and form double helix, same as parental. Yang ni conservative model dia cakap. Yang dia cakap punya satu model. Okay. Lepas tu yang ni, this is for conservative one conservative. Saya cakap conservative. Okay. Lepas tu ada another one is called semi-conservative. The technique is same. Dia macam ni, tapi dia tak akan separate. Yang ni tak akan separate. Yang ni tak akan separate. Yang ni tak akan separate. Dia terus combine will get like this. And then another string. Dia terus macam ni. Ada satu string is parental string, and another string is daughter string. They do not same that conservative. They do not dissociate again. The parental is not dissociate again. Dia terus combine saja. Dia akan terus macam ni. So that each function as template for a synthesis of new complementary string. Just like that. This is semi-conservative. So this is called semi-conservative. Okay. Another one is dispersive. So this one, each strand of both daughter molecule contain a mixture of O and duty synthesis DNA. Yang ni macam campur saja. Yang ni saya tak tahu lah macam mana dia bikin, but actually this is what they say that campur lah. Maksudnya dalam sini dia akan menjadi maybe dia macam ni. Lepas tu dia ada, lepas tu you see lepas tu dia akan macam ni. Lepas tu macam campur-campur lah. Awak pun tak tahu dia macam mana punya. Macam campur-campur sikit lah. So you use color. Macam ni lah. Campur-campur punya. Dua-dua strain ini dia campur. Campur. Ada daughter strain, ada daughter punya nucleotide, ada parental punya nucleotide. Campur-campur punya. Same as dua-dua. So you can see this. Dia campur-campur lah. Ada yang black, ada yang got dark blue, got light blue, all are they are mixed together. For this is the mixed one. So this is called dispersive. So this is the Watson and Crick. He suggest the model like this. Got three: conservative, semi-conservative, dispersive. So but he actually don't know also. Which one actually is a DNA replication model? He also don't know. He just proposed only. Okay, so in the real situation, actually only one. So the answer is actually semi-conservative. Bukan conservative, bukan dispersive. Tapi macam mana proof? Siapa proof punya? Kalau saya saja cakap, oh ini semi-conservative lah. Ada scientist akan cakap, kenapa? Who give you the brief but say that semi-conservative model is the model for DNA replication. Siapa cakap punya? Saya cakap tak boleh. Saya hanya cakap tak boleh. Ada proof punya. So, there are one type of experiment. Another experiment. Ini memang banyak eksperimen. This is the already fourth experiment. So, experiment by Melanson and Frankenstein. Ini nama mereka awak kena ingat. Kena ingat lah. Memang kena ingat punya. Kerana kalau dia keluar essay question, awak kena tulis dua nama ini. Ada satu markah. Kalau awak tak tulis, tak ada markah juga. Okay. So this experiment is determination of DNA replication model. MDC. Okay. Okay. So how he do the experiment? So he used the isotope of nitrogen. So he directly isolate the DNA of E. coli. They directly isolate the DNA of E. coli so that the the bacteria will be labeled to two isotope. One is heavy isotope. I use a different color lah. One is heavy isotope known as nitrogen 15N isotope. This is known as heavy isotope. And another one is light isotope. So this is 14N light isotope. Okay. And there are two the bacterial culture will be it will be culture immersed in these two isotope. Uh, so the firstly is immersed in the heavy isotope first. Next, after several time, it will immerse in lighter isotope. Means that the DNA now already contain now already the DNA now is incorporated into heavy and light isotope already with the nitrogen 15 nitrogen and 14 nitrogen. Okay. Okay. Then they do the centrifugation. Why we need to use the heavy isotope and light isotope? So that if after we do centrifugation, then we got a band. No, the the band, the heavy isotope, the band will be more denser. DNA band will be more denser. This is the heavy one, 15N. 15N. This one, the light one, will be more less than akan berada dekat atas punya. They have to differentiate between this. Okay. So after they do the experiment, they do the first replication first. Okay. They do the first replication. After they combine together, they do the first replication. Okay, so the first replication result give the result of hanya satu sahaja. Masuk apa? Masuk they are hybrid. Because hanya satu sahaja. Besoknya 15N and 14N ni dia sudah campur lah. Sudah campur together. Already mixed together. So for this, you can totally eliminate semi-conservative model. Because the first replication, if you do the first replication for the conservative model, then will have two band. If the conservative model is a border, then will have two band. Kalau conservative model, this is the actual condition. But if you want to prove the conservative model, actually it should be macam ni. It should be like this. 14N, 15N. It should be like this. This is conservative model. Kalau awak nak conservative model punya. Tapi, in the real situation, they buat experiment, they give the result like this. Yang ni. So, automatically, conservative model, eliminate lah. Bukan conservative model. Kadang dia tak keluar result ini. Okay, this is the first replication yang keluar. Next, it go to second replication. So, second replication yang keluar, dia dah dapat macam ni. This is lepas tu dia ada dapat ni. This one is hybrid. This one is 14N. Dia dapat result macam ni. Dia dapat result macam ni. So, means that dispersive model also eliminate. Why? Because if it is dispersive model, it will only have one band. Because dispersive model is whatever you do the second replication, even you do the second replication also you will only get one band only. Hanya akan if you dispersive model, this is the actual result. But if you do the dispersive model, you should get only one band. Hanya hybrid sahaja. You should get this result, hybrid. But tak ada. Dia tak ada bagi. Dia agak bagi ni. Satu light band, satu hybrid band. So means that dispersive result automatically eliminate. We can eliminate this result already. Because it does not contain one band. It contains two band. One is light band, one is hybrid band. So in conclusion, semi-conservative model is the model that for DNA replication. So inilah model untuk DNA replication. This is how DNA replicate. it use this model. Yang ni B punya. Yang ni means that one they separate and then got one parental strand at here and another daughter strand at here. It's same as this, also same as this. And because you see that if you use this diagram, means that this one is hybrid, this one is 14N. Yeah, 14N and also hybrid. Hybrid. Okay, sama-sama faham lah. So this is the result, same as the result of this experiment. Kalau guna eksperimen sudah proof lah bahawa semi-conservative model is true. So ya, conservative bukan, diapersis bukan. Hanya semi-conservative sahaja adalah model untuk DNA replication. So in finally conclusion, you can say that semi-conservative model act as the DNA replication model. So it is the model lah. So semi-conservative model macam mana? So actually dia macam ni. So it summarize lah, saya summarize. So it like that. The parental strand like this. Lepas tu they split into the two single DNA stranded, so it will become like that. Lepas tu ada satu daughter strand will have a daughter strand complementary to the parental strand, so it will like this. And then finally there will form single stranded double helix structure, so it will, one is like this and another one is like this. So one stranded is daughter strand, ini daughter strand, yang ni parental strand. Just like that. So this is called semi-conservative model. So only this sahaja prove that conclusion is semi-conservative model is a model of a DNA replication. Yang ada dua bukan. The others is eliminating. Okay, so actually this live is end. So next, for the live at 5 p.m., I will explain the mechanism of DNA replication. So actually this is just the briefing only, sangat senang saja. Actually, how the DNA is replicated from this to this, how it does it split, how does the nucleotide added into it, and how it is formed the double stranded. Inilah kamu akan belajar dekat DNA replication mechanism. So just like this, this slide. So thanks for your watching, and then feel free to scan this QR code. So I will see you later. I will see you later in 5 p.m. Okay, thank you.